We are going to start Unit 4. Before we get into the pathogens of the skin, I'm going to give you a little bit of an overview of what to expect during this unit, and then we'll continue on with this lecture, which will cover the anatomy of the skin, and then the pathogens and the infections that they cause. All right, so before you begin any of the lectures, take a look at the overview of Unit 4 that I posted in our Unit 4 module on our Blackboard course page. What we're going to be doing is conducting a survey of microbial pathogens and the diseases that they cause. Every time we get to a new pathogen, you're going to have to know the following 5 items. And one thing that you all are going to do is prepare your own outlines or take your own notes in this unit as opposed to filling out the outlines provided to you from myself. And so if you like making outlines, every time that we get to a new pathogen, you are going to list 5 items and you're going to look through the notes to find the information that goes with these 5 items. Usually what's given to you is the pathogen name. So this is presented to you at the beginning of each pathogen or each new infection. The next thing you all will do is determine what the cell type, or what type of, I should say, microbe type, since viruses are not cells. What type of microbe is that pathogen? Is it a bacterium? And if it's a bacterium, is it gram-positive or gram-negative? Is it a fungus? And remember, for our fungi, we have yeasts and molds, but you won't have to know which one is which in terms of identifying the pathogen as a yeast or a mold. You'll just have to know whether or not it's a fungus. Or is it a protozoan? And we learned that there were 4 different types of protozoa. To go back and review these, look at your Unit 1 lecture on protozoa. Or is it a virus? So identify your pathogen as one of these 5 major types, and then a lot of review questions will focus on the differences between these types. And that would come from your Unit 1 lectures. So some of those big picture concepts from Unit 1 were, what is the difference between prokaryotic cells and eukaryotic cells? Gram-positive and gram-negative bacteria are prokaryotes. Protozoa and fungi are eukaryotic cells, and viruses are not cells. Another big picture review topic would be, what is the difference between a cell and a virus? All right. The name of the disease that is caused by the pathogen will be another item that you have to know. And this will also be presented to you along with the pathogen name. Not just knowing the name of the disease, but the description of the disease is also important. Sometimes the description of the disease can be just a few words. For example, if you said a skin infection, that might be enough to let you know what that infection is. Or an upper respiratory system infection. Something more complex like botulism might include a more descriptive description, like the paralysis of muscles caused by the inhibition of motor neurons, for example. So when you describe what an infection or what a disease is, make sure at the very least you say what body system or what type of organ is being affected. So for example, the common cold is an upper respiratory infection or inflammation of the nose. Meningitis would be an infection of the meninges, which are the membranes surrounding the brain and spinal cord. So that would be a nervous system infection. Okay. The next item on your list that you'll have to know is whether or not the pathogen produces any of the following virulence factors. If it produces capsules, it'll say so in your lecture slides. If it is Clostridium or Bacillus, it'll produce endospores. Whether or not it says it in the lecture slides is irrelevant. You should know from Unit 1 that Clostridium and Bacillus species are two examples or two genera, genuses, that do form endospores. So if a pathogen on your list is Clostridium or Bacillus, it will form endospores. And lipopolysaccharide, or LPS for short, this is part of the outer membrane of gram-negative bacteria. So if your pathogen is a gram-negative bacterium, then it will have LPS. So if you're looking for whether or not something has endospores, look for whether or not it's Clostridium or Bacillus. If you're looking for whether or not something has or forms endotoxin or LPS, look for whether or not it's a gram-negative bacterium. All right, so these 5 major topics will be the majority of your questions, but there's going to be some additional information presented to you as well. There may be additional virulence factors, not just capsules, endospores, and lipopolysaccharide. There will be signs and symptoms. There might be some epidemiology statistics. Oftentimes there's going to be information on transmission, prevention, and treatment. So after the 5 items for each pathogen, you might want to include a 6th category as well, called additional information or extra fun facts. And then anything that seems very notable or relevant, you can keep it in that 6th item. All right, and then in addition to the pathogens, right? So there's going to be 4 to 5 pathogens per lecture. And then within those 4 to 5 pathogens, you'll need to know the 5 things and the additional info. But at the beginning of each lecture, we are going to be studying the anatomy of that body system as well, including the major parts. What are the anatomical structures that can become infected? What types of host defenses protect that body system from infection? And whether or not there are any microbes present, and if there are any notable microbes that are important as a portion of the normal microbiome, then we'll list that as well. Okay, so we are going to get ready and start today's lecture. Today, we're going to be covering these 4 pathogens of the integumentary system, which is the skin and associated organs or molecules like hair, sweat glands, etc. This week, we will also be showing the pathogens of the nervous system. So this is also this week, which is week 13 of our class. And so just a quick overview. When we talk about the pathogen, I'll tell you what the cell type is, and we'll go into a description of what these different types of infections are. So what is impetigo? What is folliculitis? One thing that you all will notice is if Staphylococcus, you have a lab on this. This is a gram-positive bacterium, or a GPC for gram-positive cocci. So we've got at least one pathogen that's gram-positive, and Clostridium is also gram-positive. It's not a cocci, so we'll just call it gram-positive. And anytime, since Clostridium species all have the same basic cell structure, that means anytime you see Clostridium on one of your list, regardless of what the species is, it's also going to be the same microbe type. So we've got 1, 2, 3, 4 pathogens on our list that are gram-positive bacteria. We have 1 pathogen on our list that's a gram-negative bacterium. So remember in Unit 1, one of our key concepts was what is the difference between gram-positive and gram-negative bacteria? So make sure you review the cell envelopes of gram-positive versus gram-negative bacteria. We have one fungal infection that we're covering, athlete's foot or trichophyton or ringworm. Trichophyton is a fungus. And we have one protozoan. We're going to cover toxoplasma this week. And then we also have a virus. So each week you'll see infections caused by a variety of different microbe types. And then in the lists that are presented to you in the Unit 4 overview, there's also the section reading where you can find information on that pathogen. So you don't have to read the entire section, just the parts that pertain to this organism right here. And so Staph aureus has information in Chapter 22, which is skin infections. It also has information in Chapter 23, which is wound infections. And so in these sections right here, we'll have bacterial and anaerobic and viral infections of the skin and of wounds, and this will be fungal infections of the skin. So again, limit your reading to just these sections and then the pathogen information within that section. All right, so now we're going to continue with just the skin pathogens, and we'll present the nervous system pathogens in a separate recording. Okay, so here's some information about the skin and the areas that can become infected. The epidermis is the outermost layer of the skin. You can see it's composed of the dead cells of the stratum corneum right here. These are the cells that contain a lot of keratin, which is a very dense, insoluble protein that helps make the skin insoluble, or it makes it a very good first line defense or barrier against pathogens getting into our body. So pathogens can't get through that keratin layer of the stratum corneum. The next layer is the dermis. And so the dermis is the middle layer. It contains a lot of the connective tissues that hold stuff together, the hair follicles, the sebaceous glands, some of the blood vessels and nerve endings. These are all found in the dermis layer, which is still a part of the skin. And then the subcutaneous layer is the deepest layer of the skin, and this is where we'll find the adipose tissue or the fat. So these yellow globs are the fat or adipose tissue. And then there's a muscle layer underneath. So we've got fat and muscle in the bottom layer of our skin. So it's important to know that skin is not just a tissue, it's not just epithelial cells. It is a mixture of all different types of tissues in these three different layers. And because of the complexity of the skin, it's referred to as an organ rather than a tissue. So if something affects just the epidermis, since this is going to be exposed to most air, a lot of aerobic bacteria or aerobic infections will happen in just the epidermis. But anaerobic infections, this will be when oxygen gets depleted. These will be anaerobic infections will be caused by either facultative anaerobes or strict anaerobes. These will occur more in the dermis or the subcutaneous layer, so they're not exposed to the air, and if an infection pierces this, the oxygen in the bloodstream will quickly get used up by aerobes or facultative anaerobes, leaving anaerobic conditions for those anaerobic bacteria to thrive and cause an infection. So anaerobic infections will be most likely to occur in the subcutaneous layer. All right, so we're going to review some first-line defenses of the skin. The first line of defense are our barrier defenses. They are innate and they are nonspecific. The primary physical barrier is the keratin itself and that stratum corneum of the epidermis. The chemical barriers or biochemical barriers include antimicrobial peptides or AMPs for short. A lot of the cells in the epithelium will secrete these antimicrobial peptides. Sebum is a lipid secreted by the sebaceous glands. So here we have the sebaceous glands next to the hair follicle. The sebaceous glands secrete sebum, and sebum has a lipid concentration which can be fermented to produce a mild acidity. It also might have a slight antimicrobial effect as well. So sebum is one chemical barrier. And then sweat, here's the sweat gland right here. The sweat obviously secretes sweat, and sweat has a high salt concentration. And when the water evaporates from the sweat, then that salt concentration is even higher, and a high salt concentration will inhibit the growth of most bacteria, except for, of course, halotolerant bacteria. Sweat also contains the enzyme lysozyme, which we covered in Unit 1 and in Unit 3. It's an enzyme that breaks down peptidoglycan, and sweat also has a mildly acidic pH, as does the fermentation of the lipids in semen, sebum. The biological barrier is the microbiome itself. So there's lots of microbes that populate the skin, and the more good microbes there are, the fewer attachment sites there will be for pathogens. Okay, our first pathogen, Staphylococcus aureus. So you have seen staph in the lab. Staph is a gram-positive staphylococci. So let's look for our 5 items that you need to know. 1 is the name of the pathogen. That's right there, Staph aureus. Remember that when we write bacterial names, the genus name is always uppercase. And then the species name right here is always lowercase. And we either italicize it or we underline it. So all Staphylococcus species are gram-positive. So gram-positive bacteria are bacterial cells. That means that they have a very thick peptidoglycan cell wall that has teichoic acids and lipoteichoic acids embedded within. And it's staphylococci. You don't have to memorize this, but it's in the name. So staphylo, if you recall from Unit 1, refers to clusters of cells, and cocci refers to circle-shaped cells. And so non-motile means the cells don't have flagella, and they don't move. All right, so that's some information about the microbe itself. The microbe type was the second thing that you needed to know. It is a gram-positive bacterium. The third piece of information are the disease names and then the disease descriptions. So this is part 3. And then you also have to know the descriptions will be coming up on the next several pages. And then finally, you need to know whether or not it produces endospores. Since it's not Bacillus or Clostridium, there will be no endospores. Since it's not gram-negative, there will be no lipopolysaccharide. And then in terms of capsules, we'll find out as we get to the virulence factor. I'll let that remain a mystery for now. Okay, so let us go through and talk about these different disease descriptions, and then we'll learn more about the virulence factors of Staph aureus that enable staph to cause such infections. Okay, the first type of infection would be hair follicle infections. So briefly, a hair follicle infection is an infection of this hair follicle right here. If an infection is limited to the hair follicle, like right here, or even a little deeper in the hair follicle, these 2 types of infections would be called folliculitis for inflammation of the hair follicle. So whenever we see that word itis, that means inflammation or infection. So we have itis for inflammation and follicu for the follicle. If the infection spreads beyond the hair follicle and there's much more pus associated with it, then it will be a more serious infection. You might even, if it is very large, it could even give you a slight fever. The book has names for these infections called furuncles and carbuncles, but we're just going to call them boils. So if you have just a folliculitis infection, you might get a small bump around the hair follicle. If you have a boil, it'll be sort of a larger zone of inflammation and infection, and there will be a lot more pus associated with it. So these two right here would be boils, whereas these two would just be folliculitis. But both of them are different types of hair follicle infections. All right, next we have impetigo. Impetigo, which is shown in this picture right here, is a lesion on the skin that often has a little bit of a crusty or a patchy appearance. This can occur followed by an insect bite, a burn, or a scrape, or it can happen around the corners of the mouth. So impetigo can be caused by staph, but it also can be a co-infection with Streptococcus pyogenes. So a co-infection is when there's more than one species present. So again, this is still a staph infection, but maybe staph plus Streptococcus pyogenes. Some of these infections can also be just caused by Streptococcus pyogenes. And then a lesion, I've mentioned that word lesion. A lesion would be any type of abnormality in the tissue on the skin, usually caused by some sort of infection, disease, or trauma. So some sort of injury or sore. And so next we have cellulitis. Cellulitis is the infection of that subcutaneous layer. So cellulite is a fatty tissue associated with the skin, and that fat tissue is found in the subcutaneous layer. So cellulitis would be an infection of the subcutaneous adipose tissue, and sometimes even the muscle underneath. So the cellulitis will be more serious and more likely to be an anaerobic infection than folliculitis. All right, next we have wound infections. So wound infections can occur if there is a wound that you get from scraping your foot or cutting, having a cut on your hand, or it can come from a surgical opening or site. So that surgical opening can be on the surface of the skin, or it can be on artificial valves or joints, like a hip replacement. So staph infections, if it really infects the inside of your body, that'll be not necessarily just associated with the skin, but more likely to become systemic or system-wide when the microbes get into your bloodstream and travel to a different region and sort of take hold. So these types of staph infections will be the most serious. They can be on the skin or other parts of the body. And if the toxins get into the bloodstream and cause a massive loss of blood pressure, this type of systemic staph infection would lead to toxic shock syndrome, or TSS. So staph is very versatile. It causes infections not just of the skin, but it can cause infections of heart valves, of internal bone joints. It can cause infections in the gastrointestinal tract. And so it's very versatile in terms of the conditions that it can tolerate, survive in, and also grow. So now we're going to get to our sixth category, which is extra information. And I have a lot of extra information about Staph aureus. Today's lecture will be largely devoted to this pathogen compared to the other three. So one thing that makes staph so adaptable is its ability to survive high salt concentrations. In Unit 2, we learned that an organism that can grow at high salt concentrations is called halotolerant. And in the lab, Staphylococcus aureus was our halotolerant microbe. And so remember, in order to be a virulence factor, it's going to participate in one of those 5 stages of infections. Halotolerance helps with transmission. If being halotolerant helps it survive longer in the environment, then that will help with transmission. But because our skin can be very salty, when sweat evaporates, if staph survives on the skin for a long period of time and then we get a cut, staph will be much more likely to gain entry into that wound. So halotolerance helps with that entry stage of causing an infection. It also is what helps it adapt to so many different conditions around the human body. Capsules, which are extracellular polysaccharide layers, we've described them as invisibility cloaks. So here's a capsule. It's a sugary coating or a polysaccharide layer that surrounds some bacterial cells. And one function of a capsule is that it aids in attachment or adherence. But capsules can also help with evading phagocytosis. So capsules play multiple roles in helping cause infection. And so one thing to note, does Staph aureus have capsules? The answer is yes, they do. It's listed right here. Along with capsules, protein A, coagulase, and leukocidin, all of these are going to be virulence factors that help Staph aureus evade our host defenses. Capsules prevent opsonization by white blood cells, and that process is required for the white blood cell or the phagocyte to attach to the bacterium before phagocytosis. And so the simple way to put it is that capsules help bacteria escape or evade phagocytosis by white blood cells. Protein A is similar in its function. It'll bind to antibodies. Remember, antibodies are produced by our B cells. And if we have an antibody, so let's say this is the antibody produced by the B cell, and staph makes a protein that attaches to the antibody, then now this antibody won't attach to staph. And if it won't attach to staph, then it prevents phagocytosis by the white blood cells that recognize antibody-marked pathogens like the macrophages. So if the antibody won't attach to the staph, then the macrophages won't recognize it. So that will prevent phagocytosis or being eaten by the macrophages. Coagulase is a protein or an exoenzyme that coagulates blood and plasma. This thickening of the blood makes it more difficult for white blood cells to get to the infected area. And so just remember that a leukocyte is a general term for all of the different white blood cells. Leukocidin is a toxin that's produced that kills neutrophils, and neutrophils are phagocytic, and they are involved in second-line defense. All right, so all of these virulence factors right here help with evasion of host defenses, and then it gives the organism a longer period of time to start growing and causing damage. The next group of virulence factors help Staph aureus cause damage. Hyaluronidase digests hyaluronic acid, which is a fiber or a glue that holds the connective tissue of that dermis layer together. Collagenase breaks down collagen, which is another fiber in the skin. So this is another connective tissue fiber. And then lipase breaks down the fats in the subcutaneous layer. So these three virulence factors right here, hyaluronidase, collagenase, and lipase, these actually are involved in digestion or digesting the skin tissue itself through exoenzymes. So these three are called exoenzymes because they're digesting macromolecules that are much larger than the bacterial cell. And superantigens is a type of antigenic variation. This results in an overstimulation of T cells. This elicits a cytokine storm or a major inflammatory event, and this in itself will result in tissue damage. And this is what happens with toxic shock syndrome when these superantigens get in our bloodstream. And then hemolysins are a broad group of toxins. Staph aureus has some hemolysins. You'll probably be looking at some of these in lab when you look at your identification of the gram-positive cocci. So hemolysins disrupt or damage red blood cells, but they can also have harmful effects on other types of cells as well. So a hemolysin would cause tissue damaging by lysing or damaging host cells. So hemolysins would be an exotoxin, whereas these three damaging compounds right here would be exoenzymes. Okay. More additional information about Staph aureus, how it's transmission, prevention, and treatment. So staph infections are commonly transmitted by direct contact with one infected individual to another, or indirectly through fomites, a doorknob, a cell phone, a towel, gym towel left on a locker room floor. And then direct contact could be if you're in a hospital and you are a patient and a member of the hospital staff has staph on their hands. Prevention of staph infections is only possible through good hygiene and sanitization and also with disinfection. Sterilization is not needed to kill staph because staph is not an endospore former. Disinfectants will work against Staphylococcus aureus. And so a disinfectant like alcohol or Lysol could be used on gym equipment after you use it. Treatment, because staph is a bacterium, you can use any of the antibiotics or antimicrobials that work against bacteria. So if you have a skin infection, you can apply a topical antibiotic, which means right to the surface of the skin. Some infections, if it gets down into the subcutaneous layer or the dermis layer, you might have to take an oral antibiotic so that the antibiotic can get into the bloodstream. But there would be a variety of antibiotic options that would work against staph. And if the infection is severe, then debridement involves the surgical removals of dead tissues. And even more severe cases might necessitate amputation. If the staph is resistant to many antibiotics, including those beta-lactam penicillins or penicillin derivatives, then we call it MRSA. So MRSA stands for methicillin-resistant Staphylococcus aureus, and you've seen this slide in Unit 3. MRSA can be any of the above types of infections that we just talked about, a wound infection, cellulitis, folliculitis, etc. And MRSA infections can be community spread or hospital acquired. So a nosocomial infection or a hospital acquired infection are synonymous. We have also called that a nosocomial infection. Other types of antibiotics can be used to treat MRSA infections. Some of these options might include tetracycline or clindamycin, vancomycin, or sulfa drugs. Vancomycin is probably the most potent of those, but some strains of MRSA are also resistant to vancomycin, and so we call them VRSA for vancomycin-resistant Staph aureus. Okay, pathogen number 2 on our list is Clostridium perfringens. So that is the name of the pathogen. That's item number 1. It is gram-positive. That's item number 2. And it causes gas gangrene or myonecrosis. So that would be item number 3. What gangrene is, it's associated, or the signature of gangrene is the blackened tissue right here. So if that blackened tissue of the muscles, if those muscles are dead and turn black, that's why we get myo for muscles, necrosis for dead, or deadening, blackening. And so what you get is a gas production in the deep layers of the skin, in the muscle layers of the skin. And then this results in the formation of this blackened necrotic tissue. And it can become fatal if not treated. So that would be our disease description, which is the 4th thing that you need to know for every pathogen. And the 5th is endospores capsules, or LPS. It is a gram-positive bacterium, so it's not going to have lipopolysaccharide. It is, however, Clostridium, so that means it does have endospores or forms endospores. And it will not form capsules. I'm not going to say that for every single pathogen that doesn't have capsules. For the future, or for all other pathogens from here on out, if you don't see capsules on the list of virulence factors, then that means the pathogen does not form capsules. Another fun fact about Clostridium perfringens, so additional information. This would be part 6. Clostridium perfringens is an obligate anaerobe. What that means is it will not grow in the presence of oxygen, and oxygen will kill it. And it grows by fermentation. It's not going to grow by alcohol or lactic acid fermentation. It's going to grow by a mixed acid type of fermentation that will produce organic acids as end products, as well as some carbon dioxide. Right, so fermentation is a type of anaerobic metabolism that usually first involves glycolysis. Then that end product from glycolysis is often reduced to something else, and we get organic end products as opposed to only CO2. But in the Clostridium, the fermentation that Clostridium perfringens does produce some CO2, and that CO2 is what causes the gas production in the muscles, and that's what causes the bubbles that you see in the skin. So that is not a good time. More extra information about Clostridium perfringens. The gas formation from fermentation is a virulence factor. It has some exoenzymes we've seen already, so we've already defined these with staph. Hyaluronidase, digests that hyaluronic acid. Collagenase digests collagen, and both of these are connective tissue fibers that are found in the skin. And Clostridium perfringens also has an alpha toxin, which is similar to a hemolysin. It is a toxin that has an effect on the cell membrane, and it results in the cell lysing or rupturing. So that it has a very damaging effect. And of course, as mentioned before, Clostridium perfringens has endospores. And so if the endospores can survive in a long period of time in the environment, so to review, an endospore is formed by Clostridium and Bacillus. It's formed within the cells. And when the vegetative cell dies, the endospore can survive. Endospores have a very tough keratin coating, and they are very tough to kill, so they survive for a long period of time. In fact, in order to kill an endospore, you usually need some sort of sterilant rather than a disinfectant. All right, so because they last for a long period of time and survive under harsh conditions, that means a gangrene infection might not be transmitted from one person to another, but instead, if we ingest some of these endospores that come from soil, or if we breathe in some of the soil and some of that gets down our throat, or if some endospores come out of our fecal material and we get some of that into our urinary tract, you could get some sort of infection from within the body, or an endogenous infection if you have some Clostridium perfringens in your GI tract. The only prevention is through cleaning wounds properly, removing dead tissues. Remember that Clostridium perfringens is an obligate anaerobe. So if our tissues aren't anaerobic, then it can't survive. And that leads us to an interesting type of treatment called oxygen therapy. If you supply O2 to the infected tissue, this will kill any types of anaerobes like Clostridium perfringens. Okay. Other types of treatments include antibiotics. I've put these in bold here, not so that you memorize what antibiotics are used, but so that we can review the mode of action. So remember that penicillin is a peptidoglycan synthesis inhibitor. And clindamycin, it is used against it. I don't know if we talked about this one in Unit 3, but a lot of the antibiotics that end in mycin, with the exception of vancomycin, inhibit 70S ribosomes. And of course, the surgical removal or the debridement of infected areas is something that also might be required if an infection gets severe, and amputation as well to prevent the infection from becoming septic. All right. Next on our list, we have the chickenpox virus, or varicella zoster, VZV for short. This is a picture of the virus right here. So the microbe type, so the name of the pathogen is it's varicella zoster. The microbe type is that it's a pathogen. Just to review a little bit about viruses, remember that viruses contain a protein coat called a capsid. And inside the virus can be a nucleic acid, and that nucleic acid can be DNA or RNA. And it can be single or double-stranded. And in this case, we've got double-stranded DNA inside. All right. And it has an envelope. Not all viruses have envelopes, but chickenpox or varicella zoster does. So the envelope is a lipid layer that surrounds the virus. It's easily removed by disinfectants and also by hand washing. So enveloped viruses tend to be more susceptible to disinfectants and hand washing. And the envelope is actually derived from the host cell's membrane. All right, so chickenpox is the infection. A description of chickenpox: it's a skin infection associated with lesions or pock marks that can last for up to 2 to 3 weeks, as you see here. The other signs and symptoms associated with the chickenpox include fever and rash. Chickenpox has a very long incubation period compared to many other viruses, which means that after 2 to 3 weeks, you may come down with chickenpox after being initially exposed to it. Chickenpox is not that serious in children, but it can be much more serious in older adults, immunocompromised individuals, or newborns. It also can reemerge later in life as shingles, which can be very, very painful. Since it's not a cell, it's not going to have endospores, capsules, or LPS. So none of the viruses will, because these are all bacterial cell structures. So anytime you have a virus on the list, you can automatically say no to those features. What viruses do have are spikes that attach to host cells. And here you see that spike attaching to a host cell. And when the virus gets into a host cell, it'll go through the lytic cycle, which results in the lysis or the death of the host cell. The virus can also be latent or dormant, and this is why it can reemerge later as shingles. So in shingles, or what happens is the virus will go latent or dormant in some of the nerve cells near the skin surface called ganglia cells. And when the virus reemerges later in your lifetime or reactivates from its suppressed state, the lytic cycle will continue right at these nerve endings where the nerve endings meet the skin. And so it's a different type of skin infection, but and it's more painful. It's more associated with the nerve endings of the skin rather than just random places in the skin. So from everything I've heard, it is not a good time. Varicella zoster or chickenpox is transmitted through respiratory droplets, which come from coughing, sneezing, or talking, or direct contact with the fluid that comes from the lesions. Sounds disgusting. It's highly contagious or highly communicable. Chickenpox and shingles are both vaccine preventable. They are preventable through an attenuated vaccine, which means they use a weakened or a modified virus. And the shingles vaccine can be given to adults that have either not had chickenpox when they were young or in a much higher dose to try to help protect you later on in life when your immune system might be a little bit lowered. There's often no treatment necessary for chickenpox or for varicella zoster viruses. The infection will usually resolve on its own. But if you are in a high-risk group, you may be prescribed an antiviral, or if you're just having a bad time, an antiviral might be prescribed. So acyclovir is the same type of antiviral drug that's given for cold sores and genital herpes. The chickenpox virus or varicella zoster does belong to the same family as the herpes viruses. The family is herpes viridae. So because it's a virus, we can't use typical antibiotics. It cannot be cured since it can become latent. These antiviral drugs don't cure an infection. They just help to reduce the severity of symptoms and reduce the amount of time it takes to recover from the infection. All right, next we have trichophyton. We don't have a single species of trichophyton that can cause athlete's foot or ringworm. So trichophyton is the genus name, and there can be a variety of different types of species. So you can just know the genus name for the pathogen name. Trichophyton is a fungus. And so if you recall the definition of a fungus, it is a microeukaryote. So eukaryotes have ADS ribosomes. They have a nucleus containing the DNA. They might have organelles as well. Fungi can be either yeasts or molds. This right here, as you can see the filaments, is an example of a mold. So molds are multicellular and filamentous. All right. And another key fact about fungi is they, unlike protozoa, they have cell walls, but they are not made of peptidoglycan. They are made of the polysaccharide chitin. All right. This fungi is a type of mold. It belongs to a group called the dermatophytes. You don't need to memorize that, but derma, like a dermatologist, usually refers to skin. So these are fungi that can colonize and live on the skin. So names for the infections could be dermatophytosis, dermato for the skin, or cutaneous. Cutaneous also refers to skin, and mycoses is the general name for fungus infection. Other common names would include athlete's foot or ringworm. And I didn't know this for a ringworm. I didn't know this for a very long time. When I was a child, ringworm terrified me because I thought it was a worm growing in your skin. But it is not a worm. It just is slightly raised and bumpy, and it's in a circle, hence the name ring. And it's not a worm. It's a fungus. So that's a little less gross than a worm infection. All right, and these infections only occur in the stratum corneum, which is that uppermost layer of our skin, which contains keratin. And keratin is also found in our hair and nails. And so these fungi will actually eat keratin found in these tissues. So it can affect your skin in various parts of the body, especially if that skin is moist or wet. And so one way to prevent these types of infections is by keeping your skin clean and dry. So keratinase is the enzyme that digests keratin. As far as the other three virulence factors go, don't confuse there's mold spores and there's bacterial endospores. So even though molds have spores, they don't have endospores. So for those three virulence factors I wanted you to know, we don't have any of them. Only bacteria potentially will have one or more of these. And transmission is only through indirect modes, maybe being exposed to some of those mold spores. Avoiding contact and good hygiene are the only form of prevention. Washing, drying, keeping your skin clean and also dry. And because it's a fungus, you can't use a topical antibiotic, but you can use a topical, which is directly applied to skin, an antifungal drug. So one of the antifungal drugs that we learned about was fluconazole. And so if it's just a skin infection, you can often buy these topical antifungals over the counter, or the oral antifungals are often prescribed. All right, that's all I have for you today on our skin infections. Hope you all have a good weekend.